Hi class, this is your instructor, Skylar Huff, and we have now made it to the 35th chapter. This is stem structure and transport. A vegetative, vascular plant has three parts. They are roots, leaves, and stems. Please bear in mind that what roots do is they anchor those plants and absorb materials from the soil, such as water and minerals. On the very same note, leaves function primarily for photosynthesis. They convert that radiant energy into chemical energy by way of a carbohydrate molecule. Stems, the namesake of this chapter, they link the plant's roots to its leaves. Stems, the namesake of this chapter, they link the plant's roots to its leaves and are usually located above ground. Although many plants have underground stems, the stem exhibits a variation of forms. They range from the rope-like vines to the massive trunks seen there up above. So what you have above, of course, is giant sequoia trees. Yes, they are giants. They are the most massive trees found on Earth. The stems here, they grow so huge to be upwards of 250 feet or more. And this photo came from Sequoia National Park. This is in California, and I can hardly wait to go on this trip. So stems can be herbaceous with soft, non-woody tissues, or they can be woody with extensive hard tissues of wood and bark. Stems perform three main functions in plants. Those functions are as follows. Stems of most species, they support leaves and those reproductive structures. The upright position of most stems and the arrangement of the leaves on them allow each leaf to absorb light for use in photosynthesis. Reproductive structures, such as flowers and fruits, are located on stems in areas accessible to insects, birds, and air currents, which transfer pollen from flower to flower and help disperse seeds and fruits. Secondly, stems provide internal transport. They conduct water and dissolved minerals, being those inorganic nutrients, from roots, where these materials are absorbed from the soil, to the leaves and other plant parts. Stems also conduct the sugar produced by leaves by way of photosynthesis to roots and other parts of the plant. Please bear in mind, however, that stems are not the only plant organs that conduct materials. The vascular system is continuous throughout all parts of the plant, and conduction occurs in roots, stems, leaves, as well as reproductive structures. So as we continue through the chapter, please keep in mind our key concepts. The primary tissues. And I'm referring to the primary tissues of stems: the epidermis, the cortex, the pith, xylem, and phloem. And these develop from that shoot apical meristem. And then we have those secondary tissues, being the wood and bark, and they develop from the two lateral meristems, being the vascular cambium and cork cambium. So let us continue on now with the 35th chapter. Stem growth and structure. So thirdly, the stems produce new living tissue. So they continue to grow throughout the plant's life, producing buds that develop into stems with new leaves and or reproductive structures. In addition to the main functions of support, conduction, and the production of new stem tissues, stems of some species are modified for sexual reproduction, or if green, to manufacture sugar by means of photosynthesis. Now on to stem growth and structure. So as we are here, just have a bit of review. Primary growth, it increases length at apical meristems by way of the root and shoot systems. Secondary growth increases girth at lateral meristems by way of course, those stems and roots. All plants, I repeat, all plants have primary growth, others of which have both primary and secondary growth. So primary growth would only be in herbaceous plants, whereas primary growth and secondary growth is found in woody plants. Please keep this in mind. So as I've stated this, the primary growth is found in all plants. So those lateral meristems, of course, will be providing the new tissues formed by the secondary tissues, meaning this is what distinguishes the primary tissue from that secondary tissue. So let's get now to the herbaceous dicot and monocot stem structure. They differ. So as we begin, what you all should do now is look closely at figure 35.1 and have along with you figure 35.2. So as I go through these, please bear in mind that I'll begin here with monocot and dicot stem structure, and I say they all contain an epidermis, or in the case of a periderm, meaning that outer tissue. Then they contain the ground tissue and vascular tissues. So I'll begin with the cross-section of a sunflower, Helianthus anus, the stem. Note the vascular bundles arranged in a circle around that central core of that pith. So as you draw this, yes, you should be drawing now. Draw your circle, and we'll begin with the epidermis. Just inside the epidermis, we have that cortex. And then just outside the cortex, we get to those vascular bundles, containing, of course, both xylem and phloem. So from the epidermis, looking closely at figure 35.1b, we have the epidermis that is there. Then is the cortex. So next up will be the phloem fiber cap. Then the phloem itself, making the vascular bundle, the vascular cambium, and then the xylem. Those are what are there. And then continuing inside, we get to further what is called the pith. And the pith is nothing more than ground tissue at that center of a dicot stem. So now that I've gone through it, this is where I was. So please keep in mind for your lecture test, you know these portions. And not just for lecture, but just as I have lecture, we also have lab. So this is Helianthus anus, the sunflower. This being where we are. So at a cross section, we now go here. So I began outside and I'll begin outside again. This is that epidermis, that outermost layer. And this would be something I would look for on the lab test. It's been on every lab test I've given. Just inside the epidermis is what is called the cortex. It's between the epidermis and what will come being the vascular bundle, or vascular bundles being plural. And then I ended things with that pith. The pith is there, and it is the ground tissue that is at the center of the dicot stem. I then went to the larger close-up of the vascular bundle. So in each bundle, we have the xylem, which is going to be located toward I put the stem's interior. So I say again, to the interior of the stem is where the xylem is found. And then to the outer, toward the exterior of the stem, is where the phloem is found. So the phloem is found right there. So that is what's contained within the vascular bundle. So I say again, it's the phloem fiber cap, the phloem itself, the vascular cambium, the xylem, and of course what's within being that vessel element. To the outside is that cortex, and then ultimately the epidermis. This is a dicot or Eudicot stem. Next up, the monocot stem. So the monocot has things that are quite different. And what is different is that there are no lateral meristems here because it has no secondary growth. It also is, I repeat, no distinct cortex and pith found here. So here, what we're going to draw is going to be from figure 35.2, and it's called the cross-section of corn, Zea maize, the stem. So this should show those scattered vascular bundles. I say this is what makes it very unique. This should be one of the easier things, I say, to grasp. But if it's not as easy to grasp, I say please study well and ask me any questions that you all might have. So as we look at it here together, so you should draw your circle. And we'll begin labeling from outside in. So label the epidermis. After the epidermis, please draw those scattered vascular bundles. And then, of course, the ground tissue within. That is, of course, what I call the monocot stem cross-section. So here it is for you to see. We'll do it together. So this, of course, is showing you what it is. So this is corn, Zea maize. If you look closely, those are the vascular bundles that are there that are scattered. So next up, we'll begin, as I began moments ago, with the epidermis, that outermost portion here. So just inside the epidermis, they begin, called the vascular bundles. So now they're scattered all around as opposed to being in a concentric ring as we saw in the Eudicots. So now I'll go over to figure 35-2b for that close-up of the vascular bundle here in a monocot. So yet and still, it's not going to be an epidermis that's here flanking that exterior because the vascular bundles here are scattered. So it's ground tissue that surrounds. So the ground tissue that's here, it is what surrounds those scattered vascular bundles. So first things first, this is the phloem that's here. Along with the phloem, there is a sieve tube element, one of those cells. And also right there with it is a companion cell. You all should remember all of this from the 33rd chapter. Continuing along, we now have the xylem, which is there, and then the vessel element. And then is the air space. So the bundle sheath cells are what surrounds the vascular bundles here in the monocots. So this is something I would know for the test as well as the lab exam. You've now been told. I'll nextly continue on with where we're going with the chapter. So now we'll get to woody plants. In woody plants, the Eudicots, they undergo secondary growth, and it's primarily because of what is called the vascular cambium and Cork cambium. So as I am here, keep in mind, the lateral meristems are what are responsible for this. And there are two. They're called the vascular cambium, I say again, and Cork cambium. So the vascular cambium gives rise to the secondary xylem and secondary phloem, which replaces the primary tissues both positionally and in function. So I must ask, what is transported by each? Note, if you cannot answer this question, I recommend you go back to chapter 33 and review quite well. And I asked, what is transported by each? I'll now ask, where? These two small questions, one may say, oh Huff, that's an easy question, perfect. But then someone else may say, I cannot answer the question at all. So if you're someone who cannot answer the question at all, you should very well go back and answer the questions upon reviewing the 33rd chapter. I'll now continue. Though secondary xylem and secondary phloem, they transport water, minerals, and organics vertically. We now have what are called rays. And I mention rays here because what rays do is transport those laterally. It's very important to get this because we have this tree, this dicot, whatever species it may be. Maybe it's apple, maybe it's hickory, maybe it's even pecan. So as it increases growth, and of course I say increasing growth, I guess I'll say in girth, it must have a way to transport these things laterally. So this is where rays come into play. So the materials must, I repeat, these materials must move horizontally. So rays are nothing more than chains of parenchyma cells that radiate from the center of the woody stem or root. So the vascular cambium forms rays which are often continuous from the secondary xylem to the secondary phloem. So the water and minerals are transported laterally through rays from the secondary xylem to the secondary phloem, and likewise rays form pathways for the lateral transport of dissolved sugar from the secondary phloem to the secondary xylem, and of waste products to the center or heart of the tree, which we'll get to hardwood a bit later. So I'll now get to what is called the Cork cambium, the second of two. The Cork cambium produces the periderm, which is what will replace the epidermis. Again, I say, the Cork cambium produces the periderm, which will be what replaces the epidermis. And it's happening from those parenchyma cells. So here I say it's coated with suberin, which is what is that waterproofer. And as I go through this, please bear in mind, or please bear in mind that there are figures that accompany what I'm doing right now, and especially I say figure 35-5. That is an amazing figure. We'll get to that in just a moment here together. So stomata, they are then replaced. Stomata, they are then replaced, and you're seeing here visually the development of secondary growth. So in the beginning, we have the primary xylem that is there shown in a bit of a pink. The primary xylem shown there in a bit of pink. And of course that primary phloem shown there in a bit of a teal color. And the cortex it surrounds. So if you look even closer, you will then see the remnants of that. And then you're seeing in that now, I guess I'll say kind of a salmon color, you're seeing that secondary phloem. And the secondary xylem, excuse me. The secondary xylem in the salmon color, and the secondary phloem in that lighter blue color. And then, of course, you're seeing it being replaced, not only in, I guess I'll say, position, but also in function. So there it is. So the periderm, which is there, that outer bark, shows now the remnants of the primary phloem, the cortex, and the epidermis are gradually crushed, torn apart, sloughed off, destroyed by these, of course, secondary tissues. So now, I mentioned rays already, being those chains of parenchyma cells. Well, now I'll get to the photo I mentioned moments ago. So this is Tilia. It's called basswood. Make sure you review this. This is Tilia. It's called basswood. So in this figure, a lot is being shown. So at the bottom, in the center, we have the remnants of those primary tissues still remaining. Remnants of primary tissues still remaining. And with this, what we have, I guess I'll go to the outside first. There is the periderm that's there with the remnants still of the primary tissues. So this is that periderm, the bark. And then just inside the periderm is where we get to that secondary phloem. That secondary phloem. You may wonder why I spend so much time here. You will see this on your test. This has been on both the lecture and lab tests. Along the same note, here's that thin cylinder. The thin cylinder I refer to now is that vascular cambium. Please go back and review the 33rd chapter. That is your basis for understanding if you're not quite following what I'm doing here. So that thin vascular cylinder that's there, and then what follows within would be the secondary xylem. And it does say 3 years is growth. So this is the third year, second year, and of course, ultimately, the first year of growth. This is that secondary xylem. And one thing that I have yet to mention now are those rays. So if you look closely here, and if you need to, turn your head sideways. These are the rays. So in this figure, it appears to be a kind of, I guess I'll say, gold color. So there it is, able to connect both of which, the secondary xylem and secondary phloem, and transport the products laterally throughout that plant. It's so amazing to see these. So amazing to see these. Back to where I left off. So as we are here, the periderm I mentioned moments ago is what replaces the epidermis. And those cork cells, which are dead at maturity, they have that waterproof barrier. The waterproof barrier, of course, is because of the coat with suberin. And the cork parenchyma cells also store a bit of water and starch granules. So now I mentioned just moments ago, stomata are replaced by lenticels. I repeat, stomata are replaced by lenticels. What lenticels do, class, is, of course, allow gas exchange through the periderm. So if you closely look here at figure 35.6, this is it. That is that lenticel, and that lenticel is there primarily to allow gas to be exchanged. It's so amazing how this is happening. Much like, of course, you all saw with the stomata before. So what I'll get to now is, I'll get to wood as described by plant structure. Wood as described based on plant structure. So if you have ever examined types of lumber, you may have noticed that some trees have wood with two different colors. You may have noticed such a thing. And I'll show you another figure that will exhibit this, but not quite yet. So the functional secondary xylem, meaning the part that conducts water and the dissolved minerals, is called sapwood. Here we are. It's called sapwood. And to help you with that, an easy way of doing this is sapwood is the functional secondary xylem. It's thin, it's younger, and lighter in color, and it's also going to be found closer to the bark. Up next, I'll go to heartwood. So heartwood is that older wood in the center of the tree, typically brownish red. So with that, I'll say that heartwood is no longer functional. It's a storage site for waste products. So I say because it's denser than sapwood, the heartwood provides the structural support for trees. So evidence also suggests that the heartwood is also more resistant to decay than is sapwood. Up next, I'll contrast the two of which being softwood and hardwood. So softwood is found from conifers, meaning you're saying, softwood I am. One day you all will buy a home, and when you do, just make sure you know that those cone-bearing gymnosperms are those that bear softwood. So it's because they lack fibers and vessel elements, meaning the wood of the pine, I'll just be blunt, typically lacks those fibers, meaning with those thicker secondary cell walls. So the conducting cells in gymnosperms are tracheates. Hence, these cells, in their differences, generally make the conifer wood softer than wood of flowering plants. So up next, I'll get on down to hardwood. To help you out and make it easy, hardwood class. Hardwood is from flowering plants. I mentioned some of those just a moment ago, such as hickory, or even the apple, and even pecan. So that's why I use those, of course, when I like to smoke my meat. And it's most amazing when it's smoked. So next up, of course, is going to be to contrast both springwood and summerwood. So springwood would be that also thing called earlywood. So as it's stated here, springwood or earlywood has those large vascular cells when water is plentiful. They say there are sometimes things called spring showers. Well, thereafter is summerwood or late wood. So this wood is forming, of course, thereafter. And it has narrower conducting cells, and this is when the water is less plentiful. And this is easily discernible. And when I say easily discernible, I'll go back to where I was with figure 35.5 to show you such. So look closely, class. If you look here, this is nothing but spring wood where my mouse is. If you look closely, see how large those cells are? To contrast that with the smaller cells in this area where the mouse is. And that would most definitely be summer wood. Summers are typically warmer, and of course, with less precipitation, typically. So I'll get now back to the woody twig. So those twigs, of course, have those buds, also known as embryonic shoots. So with this, the terminal bud is also always going to be at the tip of that stem and protected by that outer bud scales. So there also will be axillary or lateral buds. I call them axillary buds, located at the axis of the plant's leaves. And I said the location of the axillary bud is your key to determining whether, of course, its leaf form, I repeat, that is your key to leaf form as far as it being simple or compound. Please go back and review that from the 34th chapter and make sure you have taken notes. It is on the test, the lab test especially. So when terminal and axillary buds grow, they form branches that bear, of course, leaves and or flowers. It's amazing to see, so please walk outdoors when you have the opportunity and look at this. View it. So during winter dormancy, the bud scales that cover that terminal bud, they protect that delicate apical meristem. It's amazing how temperatures can fluctuate from season to season. And as the temperatures do warm up, eventually there will be, of course, what comes thereafter. So each spring, when the bud resumes growth, those bud scales fall off, leaving bud scale scars. And that same is the case, of course, when you have the leaf scar, meaning a leaf scar shows where a leaf was attached to the stem, and within the leaf scar, you can also see, of course, the vascular tissue by way of those bundle scars. Make it plain. To make everything plain that I've just done, if you look closely, this is that terminal bud. It says it there. This is a terminal bud. And it has those bud scales that protect those developing or at least delicate tissues within. In the springtime, this is where you all will see those leaves. What we now have is going to be from what was once a terminal bud here. So it says terminal bud scale scars to this terminal bud scale. Well, the terminal bud with bud scales, excuse me. That is 1 year's growth. So what I'm getting to here is the node is a point of attachment for what was once a leaf. And from node to node, I'll repeat, from node to node relieves the internode. I'll get even more specific now. So what we also have is an axillary bud here. With that, axillary buds are always going to be in the leaf axial. So yes, there was a leaf that was there last season. So now we have that leaf scar. And along with that leaf scar, as was just stated to you, I repeat, along with that leaf scar, as with what was just stated, are those bundle scars. So you can see those bundle scars being labeled there. And that was for the vascular bundles. So I've done that with you. And I guess I'll say with this, keep in mind these things, especially for lab. So next to which, I'll get to, of course, the photo, figure 35.8. I mentioned this earlier. You can see a stark difference in even the color. So you have that sapwood being that outer portion here. The sapwood that's there, that lighter portion nearer to the bark. And then you have what's within being that heartwood, the heartwood that's here. So this shows that wood of older trees consisting of that dense central heartwood and that outer sapwood layer. So keeping it in mind, the sapwood has a functioning xylem that conducts water and dissolved minerals. And those annual rings in the heartwood are very, I say again, they are very conspicuous. But I would never ask you all to count those rings that you see there. Continuing on. So with the tree rings here, this is used in a number of ways. So the difference between summer wood of 1 year and spring wood the following years gives us those annual rings. If you don't know what I mean, there it is again. That's where the annual rings come from, class. So the age of a woody stem in temperate zones, I repeat, in temperate zones, can be determined by annual rings. And the rings cannot be used if, of course, the tree is a tropical tree. I'll get now to the tree ring analysis very briefly. So the size of each of those rings depends on those local weather conditions. So it just depends, of course, on how much precipitation that was there. The study of this is something that you could study in ecology, anthropology, environmental science, or even geology. So if this interests you, I would say please do pursue it. And if I can help you, please let me know. I am now at water transport. Please keep in mind with water transport class, you will be tasked with tracing the path of a drop of water through a plant on your final exam. Yes, that's homework. I repeat, what you should be able to do is be able to trace the path of water. I repeat, the path of a drop of water throughout a plant. So we'll begin this here, and we'll finish this in the next chapter, more specifically. So roots obtain water and dissolve minerals from the soil, and these materials then transported up. They're transported upward class to leaves, by way of the stems, to flowers, fruits, and seeds. On the converse, sugar molecules are translocated. I say again, on the converse, sugar molecules are translocated in phloem from their place of manufacture, or the breakdown of starch, throughout the plant body, including into the subterranean roots. So to break that down just a bit, water and minerals are transported to other parts of the plant via the xylem. Dissolved sugar is translocated via the phloem. I can't break it down any more than that. Please, class, contact me with questions. Here we are. So the xylem transports water and minerals up. And I mentioned just moments ago, the phloem, it translocates sugars up or down in separate phloem cells. That's the major thing to get, class. Water transport, it begins, and as I say this, you can use a flow chart just like the one in your text, seen on page 755. You can use a flow chart with arrows to do this. That's what I'll do. Water transport, from soil to roots. I'll repeat, water transport, from the soil to the roots, and thereafter into the tracheids. From those tracheids, it moves into the vessel members. And what I'm getting to now is the root xylem, class. The root xylem. So I'll start it over and say it this way to make it, I guess I'll say, a bit less specific by going from the soil to the root tissues. From the root tissues into the root xylem, to the stem xylem, the leaf xylem, ultimately into the leaf mesophyll, and then ultimately into the atmosphere. If you have not remembered, that would be the process of transpiration, loss of water by aerial parts of the plant. So this is, I say, similar to drinking water through a straw. I say again, this would be similar, class, to drinking water through a straw. No energy is required. There is no energy expenditure, class, for this process to occur. It's a natural physical process. So with this being the case, as this water is moved, water initially moves horizontally into the roots from the soil, passing through several tissues until it reaches the xylem. Once the water moves into the tracheids and vessel elements of the root xylem, it travels upward through a continuous network of these hollow, dead cells from root to stem to leaf. So those are all minerals. They're carried along passively in the water. So as I mentioned, there is no energy expenditure of the plant itself. It just moves as a result of a natural physical process. So the transportation of xylem sap is the most rapid of any movement of materials in plants. And as I say this, it's pretty fast. I mean, in a herbaceous vine, the maximum rate of movement in xylem can be up to 250 centimeters per minute. And in conifers, it's a lot slower, 2 centimeters per minute. And in woody dicots, 73 centimeters per minute. And in the herbaceous Eudicot, or even monocot, 100 centimeters per minute. So if you ask, how does it water move to the tops of plants, such as from those over 250 foot tall giant sequoias at Sequoia National Park in California? It's either pushed up from the bottom of the plant or pulled up by the top of the plant. So although plants use both mechanisms, current evidence indicates that most transported water through plant is pulled to the top of the plant. So it's pretty amazing how this has happened. And it's explained by that difference in water potential, going all the way back to your bases with osmosis. That's where it is. So it's happened by looking at water potential. It is just simply the free energy of water So this goes back to chapter 7 of which, when you got to your introduction to how things move. So water potential, represented by psi, is an important in plant physiology because it is a measure of the cell's ability to absorb water by osmosis. So osmosis, that diffusion that it is, involves the net movement of water through a selectively permeable membrane from a region of higher concentration of water to a region of lower concentration of water. So water potential also provides a measure of the water's tendency to evaporate from cells. So the water potential of pure water is 0, meaning it's 0 megapascals, because it cannot be measured directly. And if you've never heard of the megapascal, it's a unit of pressure equal to about 10.2 kilograms per centimeter squared. However, botanists can measure differences in free energy of water molecules in different situations when solutes dissolve in water. The free energy of water decreases. Solutes induce hydration, in which water molecules surround ions and polar molecules, keeping them in dissolution by preventing them from coming together. So the association of water molecules with hydrated molecules and ions reduces the motion of water molecules, decreasing their free energy. Thus, here it is, dissolved solutes lower the water potential to a negative number. So water moves from a region of higher or less negative water potential to a region of lower or more negative water potential. So from here, I'll keep on moving. And this is, I say, quite amazing how it's happening, that natural physical process. And I won't spend much time on that pressure flow model to explain translocation, but let's now begin the translocation of sugar. Let us begin. The translocation of sugar. So here, I'll use sucrose. It is that translocated form and is from source to sink, such as the root being an example of a sink, the apical meristem being an example of a sink, a fruit, or even seed. Those are 4 different examples of sinks. I'm stating this because on your test, you will not only trace the path of a molecule of water throughout a plant, but you also will trace the path of sugar throughout a plant from source to sink. And please do choose a sink. I just gave you 4 examples of sinks. Be specific, please. Again, I say, please be specific. Sucrose, the disaccharide made up of glucose and fructose, is produced via photosynthesis and then carried. So phloem translocation is not as fast as xylem transport. So again, sucrose, as it's produced via photosynthesis, is carried in the phloem, and the translocation of sugars via phloem is not nearly as fast as xylem transport. So it moves up and down from sieve tube cell. The source, it moves, class, up and down from sieve tube cell, the source, to a sink, i.e. some seed, fruit, root, or apical meristem. And the way this happens, in contrast to the way it happens so passively with water, it happens by a way of active transport, requiring adenosine triphosphate, known as ATP. So what happens first is the sugar molecules must firstly accumulate in the sieve tube element. So as the sieve tube element gets that sugar, the sugar is moving into that cell actively. I repeat, by way of active transport, sugars enter into the sieve tube element. So next, of course, will be that water diffuses into the sieve tube element. I say again, water moves in by way of osmosis from the xylem cell into that seed development. So here we are, the leaf cell, the source. So here we have in red the sugar. Here. The sugar, I say again, moves into that sieve tube cell by way of active transport. The second part, with my arrow, I hope you all are writing this out as I mentioned earlier. You can use arrows, use a flow chart. The second portion here is that water, by way of osmosis, diffuses into the sieve tube cell, that sieve tube element. So next of which is this increases turgor pressure. This takes you back to your first biology course, Biology 1, where you learn that turgor pressure is that thing that plant cells have when they are normal. Meaning a normal plant cell is in a hypotonic environment, and those cells are turgid. So that increases the turgor pressure, so now the sugar leaves the sieve tube element at number 3. The sugar now leaves the sieve tube element. So the first thing first was sugar moved into, I repeat, start back at 1. The sugar moved into the sieve tube element by way of active transport. Water then diffused into that sieve tube element, increasing turgor pressure. Now sugar leaves the sieve tube element. Along with it, there's a second blue arrow there because water also leaves, diffusing into the xylem. Water also leaves, diffusing back into the xylem. So as that sugar then leaves, the sugar then leaves and goes to some sink. Notice we began with a source. Now we're in a sink. Please choose your sink wisely. From source to sink, I say. So in this case, that sink is a root cell. This could be in a potato plant, because they say those potatoes are storage roots. This has been the 35th chapter. Please review this. Please read this. Because this is the way in which it happens. For instance, if you're wondering more about what's going on, current experimental evidence that supports translocation of dissolved sugar in the phloem is by the pressure flow model. And this was back from 1926 by Munich, the scientist. So the pressure flow model states that solutes, meaning each of those dissolved sugars, they move in phloem by means of a pressure gradient, that is, a difference in pressure. So the pressure gradient exists between the source, i.e., where the sugar is loaded into the phloem, and the sink, where the sugar is removed from the phloem. So at the source, the dissolved sucrose is moved from a leaf's mesophyll cells, where it was manufactured, into companion cells, which load sucrose into the sieve tube elements of the phloem. Sucrose loading occurs by active transport. I just told you all this. I say again, sugar loading occurs by active transport, a process that requires ATP, adenosine triphosphate. So the ATP supplies energy to pump those protons out of the sieve tube elements, producing a proton gradient that drive the uptake of sugar through specific channels by co-transport of protons back in the sieve tube elements. So in co-transport, in this system, it's involved with phloem loading, meaning sugar is moved from a region of low concentration to a region of high concentration by coupling its transport to the transport of protons down the concentration gradient. Hence, the sugar accumulates in the sieve tube element, and of course the increase in dissolved sugar in the sieve tube element at the source, a concentration that is 2 times or even 3 times as great at the surrounding cells, decreases, makes it more negative, and the water potential of that cell. As a result, the water moves by osmosis to nearby xylem cells into the sieve tubes, increasing the turgor pressure, increasing the hydrostatic pressure inside them. Thus, phloem loading at the source was given. At the sink, the sugar is unloaded by various mechanisms, both active and passive transport, and with the loss of sugar, the water potential in the deciduous element at the sink increases. It becomes less negative. Hence, water moves out by osmosis to the surrounding cells. So most of this water diffuses back into the xylem and being transported upward yet again. So there you have it, class. So please do study well, class. And if I were you, I would use the figure as it is shown, this key point here, because you will write this out in your test, just as you will write out, of course, the way in which water is transported. So seeing the key point class of figure 3511, as well as the key point class on 3512. So thank you all for listening, class. This has been your instructor, Scholar Huff. If you need me, class, as always, please email me, call my office telephone number, or see me before and or after class. Thank you all.